Hi, this is Dr. Cassandra Quave, and in today's lecture we'll be covering Chapter 11 from your textbook, discussing eukaryotic microorganisms and parasites. So thus far we've really devoted most of our time to the study of the domains bacteria and archaea, and also to viruses. However, there are some members of the domain eukarya that are also of great interest to microbiologists. The kingdoms Protista and fungi in particular contain large numbers of microscopic species. The kingdom Animalia contains helminths that can cause disease and arthropods that cause or transmit diseases. So in this chapter we'll start going over some of the principles of parasitology as they relate to these organisms. Now, there's a lot of new vocabulary that you'll need to become familiar with. First, you should know that a parasite is an organism that lives at the expense of another organism, which is called the host. Now, parasites can vary in the degree of damage they inflict on their hosts. Although some can cause little harm, others can cause moderate to severe damage. Parasites that cause disease are called pathogens. And parasitology is the study of parasites. Now, parasites also play an important role in the worldwide economy. So, for example, less than half of the world's cultivatable land is under cultivation, mostly because parasites that are endemic or always present in these lands can prevent humans and domesticated animals from inhabiting some of them. Also, parasitic infections in wild and domestic animals provide sources of human infection and can cause debilitation and death among animals and humans. Now, parasites can be divided into ectoparasites, like ticks and lice, which live on the surface of other organisms, and endoparasites, which include protozoa and worms, which live within the bodies of other organisms. Most parasites are obligate parasites, meaning that they must spend at least some of their life cycle inside or on a host. A few parasites are known to be facultative parasites, meaning that they are normally free-living, but they can obtain nutrients from a host. Hosts that are invaded by parasites usually lack the effective defenses against them, and so diseases of this type can be serious and sometimes fatal. Parasites are also categorized according to the duration of their association with the host. For example, permanent parasites, like tapeworms, will remain in or on the host once they have invaded it. Temporary parasites, however, can feed on and then leave their hosts. Examples of this include biting insects. Accidental parasites will invade an organism other than their normal host. So, for example, ticks that ordinarily attach to dogs or wild animals can also sometimes attach to humans, and the ticks are the accidental parasites in that case. Now, hyperparasitism refers to the parasite itself having parasites. So this is, again, an extra level of parasitism. For example, some mosquitoes, which are temporary parasites, can also harbor the malaria parasite or other parasites, and these insects serve as vectors. And again, vectors are agents of transmission, and this is relevant to many different human parasitic diseases. Now, an organism that transfers a parasite to a new host is known as a vector, and a vector in which the parasite goes through part of its life cycle is known to be the biological vector. The malaria mosquito is both a host and a biological vector. Now, this is in contrast to a mechanical vector. A mechanical vector is one in which the parasite does not go through any part of its life cycle during transit. Examples of this can include flies that carry parasite eggs, bacteria, or viruses from feces to human food. These are mechanical vectors. Now, regarding host types, hosts can be classified as definitive hosts if they harbor a parasite while it reproduces sexually. They are known to be an intermediate host if they harbor the parasite during some other developmental stages. The mosquito is the definitive host for the malaria parasite because that parasite reproduces sexually inside the mosquito. The human is the intermediate host, even though the humans suffer greater damage from the parasite. Now, reservoir hosts are infected organisms that make parasites available for transmission to other hosts. Reservoir hosts for human parasitic diseases are typically found in wild or domestic animals. And host specificity is something that refers to the range of different hosts in which a parasite can mature. Some hosts are highly host-specific, meaning that they can mature in only one host, whereas others may be able to mature in multiple hosts. Now, parasites also have a number of evasion strategies that they have evolved in order to become less injurious to their host and evade, again, kind of immune clearance from the host. This arrangement preserves the host so that the parasites are guaranteed a continuous supply of nutrients. A parasite that destroys its host also destroys its own means of support. So the adjustment of parasites and hosts to one another is closely related to the host defense mechanisms. Many parasites have one or more of the following mechanisms to evade the host defense mechanism. First is encystment. Encystment involves the formation of an outer covering that protects against unfavorable environmental conditions. These resistant cyst stages also can sometimes provide a site for internal reorganization of the organism and cell division. They can also help a parasite to attach to a host, or can serve to transmit a parasite from one host to another. Another means of evading the host's immune response is that some parasites can change their surface antigens. And if you recall, antigens are molecules that elicit immunity. What they do is they will actually change these antigens faster than the host can make the new antibodies. And again, antibodies are the molecules that recognize and attack the antigens. Another evasion mechanism involves causing the host immune system to make antibodies that cannot react with the parasite's antigens. A third kind of evasion strategy involves the invasion of host cells, where the parasites are out of the reach of the host defense mechanisms. When parasites can successfully evade all the host defenses, they can also cause several kinds of damage. All parasites rob their hosts of nutrients, and some can take such a large share of the nutrients or damage the hosts so that much of the surface area of the host's intestines to the point in which the host receives little nourishment. Also, many parasites can cause significant trauma and damage to the host tissues, leaving open sores on the skin or destroying cells and tissues and organs. They can also clog and damage blood vessels and may even cause internal hemorrhages. Parasites that do not evade the defense mechanisms can sometimes trigger severe inflammatory and immunological reactions. An example of this is that treatment to rid human hosts of some worm infections will actually kill the worms. However, the toxins from the dead worms that are released upon their death will actually cause more damage to the host tissue than the living parasites do. A great example of this is the dog heartworm. It can perforate the heart wall and leave holes in the heart when the worms die and decay. So it's important for a veterinarian to test all dogs for the presence of heartworms before giving preventive heartworm medication. And here you can see again just an outline of what I just covered regarding some of their different evasion mechanisms and the damage that they can cause. Again, parasites can monopolize nutrients in the host, causing nutritional deprivation in the host itself. They can also cause direct trauma and obstruction to the host tissues and vessels, and they can also instigate severe inflammatory or immunological reactions in the host. Now we're going to move on to discuss protists. Protists are members of the kingdom Protista, and this includes a diverse assortment of organisms that share certain common characteristics. Protists are unicellular. They are eukaryotic organisms with cells that have true nuclei and membrane-enclosed organelles. Although most of the protists are microscopic, they can vary in diameter from 5 microns to 5 millimeters. Now, protists are also significant from a geological and medical standpoint. For example, protists are a key part of the food chain. Autotrophic protists can capture energy from sunlight, and some heterotrophic protists ingest autotrophs from other heterotrophs. Others can decompose or digest dead organic matter, which is then recycled to living organisms. Protists also serve as food to higher level consumers. Ultimately, some of the energy originally captured by protists will reach humans. For example, energy from the sun is transferred to protists. Protists are then eaten by clams, and the clams are eaten by humans. Protists can be economically beneficial or detrimental. Some protists have tests or shells of calcium carbonate, and carbonate shells are deposited in great numbers by protists that live in ancient oceans. These, for example, form the White Cliffs of Dover, England, and the limestone used in building the pyramids of Egypt. Now, as with regards to medical significance, some protists are also parasitic, and they can cause debilitating disease in large numbers of people and even cause death, especially in poor countries that lacked the resources to eradicate the protists. Some examples of parasitic diseases caused by protozoa include amoebic dysentery, malaria, sleeping sickness, leishmaniasis, and toxoplasmosis. Now, regarding the classification of protists, like all living things, the protists display great variation, which provides the basis for dividing the kingdom Protista into sections and phyla. The plant-like protists have chloroplasts, and they live in moist, sunny environments, and examples of this include things like euglenoids, diatoms, and dinoflagellates. All of the plant-like protists, again, or algae, have chloroplasts and carry on photosynthesis. Most of them have cell walls and one or two flagella which allow them to move. The euglenoids usually have a single flagellum and a pigmented eye spot called stigma. And here you can see on the upper right side a picture of a euglenoid. In the bottom left, you'll see a picture of a dinoflagellate. Now, a dinoflagellate, these are plant-like protists that usually have two flagella, one extending behind the organism like a tail, and the other lying in a transverse groove. These are small organisms that may or may not have a cell wall. Some have a theca, or a tightly affixed secreted layer that typically contains cellulose. Now, another group of plant-like protists can have other pigments in addition to chlorophyll, and these usually have cell walls surrounded by a loosely attached secreted test that contains silicon or calcium carbonate, and most of these reproduce by binary fission. This includes the diatoms, which lack flagella, and several other groups which have flagella and are distinguished by their yellow and brown pigments. Diatoms are an especially numerous group and are important as producers of in both freshwater and marine environments. Now, the fungus-like protist, or water molds and slime molds, have some characteristics of fungi and some of animals. The water molds and related protists that cause mildew are sometimes classified as fungi. These molds, mildews, and plant blights produce flagellated spores called zoospores during their asexual reproduction, and they enlarge motile gametes during sexual reproduction. The most prominent phase of their life cycle consists of diploid cells from the union of gametes. And these protists live in freshwater or as plant parasites. They can cause such diseases as downy mildew on grapes and sugar beets and blight on potatoes. Now, slime molds are commonly found as glistening viscous masses of slime on rotting logs. They also live on other types of decaying matter or in soil. Most of the slime molds are known to be saprophytes, Saprophytes are organisms that feed on dead or decaying matter. Now, there are different types of slime molds. The plasmodial slime molds will form a multinucleate amoeboid mass called a plasmodium, which can move about slowly and phagocytize dead matter. Sometimes a plasmodium will stop moving and form a fruiting body, and each of these fruiting bodies develops sporangia, or sacs that make spores. When the spores are released, they germinate into flagellated gametes. The two gametes fuse, lose their flagella, and form a new plasmodium. Now, the cellular slime molds produce pseudoplasmodia, fruiting bodies, and spores with characteristics that are quite different from those of the plasmodial slime molds. A pseudoplasmodium is a slightly motile aggregation of cells that produces fruiting bodies, which in turn produce spores. And these spores germinate into amoeboid phagocytic cells that divide repeatedly, producing more independent amoeboid cells. Now, with regards to the animal-like protists, or protozoa, these are heterotrophic, mostly unicellular organisms, but a few can form colonies. Most of them are free-living. Some of them can be commensals, which live in or on other organisms without causing them harm, and a few are parasites. The parasitic protozoa are of the most interest to the health sciences. Many protozoa live in watery environments and encyst when conditions are not favorable. And some protozoa are even protected by a tough outer pellicle. Many of these are motile and are further classified on the basis of their means of locomotion. In this chapter, we'll cover the Mastigophora, the Sarcodina, the Apicomplexa, and Ciliata. Now, the Mastigophorans have flagella. A few of their species are free-living in either fresh or salt water. However, most of them live in symbiotic relationships with plants and animals. Some examples of Mastigophorans that will parasitize humans include members of the genera Trypanosoma, Leishmania, Giardia, and Trichomonas. Trypanosomes are responsible for African sleeping sickness. Leishmaniasis causes skin lesions or systemic disease with fever. Giardias will cause diarrhea, and trichomonads will cause vaginal inflammation. Now, the amoebozoa, also known as the sarcodines, move by means of pseudopodia. A few amoebozoa have flagella at some stage of their life cycle, and they feed mainly on other microorganisms, including other protozoa and small algae. The amoebozoa include the foraminiferans and radiolarians that have shells and are found mainly in marine environments, and amoebas which have no shells and are typically parasitic. There are a number of species of amoebas that are capable of inhabiting the human intestinal tract, and most of them will form cysts that help them withstand the adverse conditions. Some of the most commonly observed genera include Entamoeba, Dientamoeba, Entolimax, and Iodamoeba, and these cause amoebic dysenteries of varying degrees of severity. Now, the apicomplexans, or sporozoans, are parasitic and immobile. The enzymes present in groups of organelles at the tips of their cells can digest their way inside of host cells, giving them the name apicomplexa. These parasites typically have complex life cycles. An important example in the life cycle of the malaria parasite, or Plasmodium, which requires both a human and a mosquito host. The parasites which are present as sporozoites in the salivary glands of an infected mosquito will enter the human blood through the mosquito's bite. The sporozoites then migrate to the liver and become merozoites. After about 10 days, they then emerge into the blood, invade red blood cells, and become trophozoites. Here, trophozoites will reproduce asexually, producing many more merozoites, which are then released into the blood by the rupture of the blood cell. multiplication and release of merozoites is repeated several times during a bout of malaria. Some merozoites can enter the sexual reproductive phase and become gametocytes, or male and female sex cells. So when a mosquito takes a blood meal from an infected human, it can also take in gametocytes, most of which will then mature and unite to form zygotes in the lining of the mosquito's stomach. Zygotes then pass through the stomach wall and produce sporozoites, which eventually make their way into the salivary glands. And here in the picture you can see an example of Plasmodium vivax found inside red blood cells. And again, this is an apicomplexan. Now another apicomplexan, or known as toxoplasma, can cause lymphatic infections and blindness in adults and severe neurological damage to fetuses of infected pregnant women. It has also recently been implicated as a possible cause of schizophrenia. Contact with infected domestic cats and their feces, and consumption of contaminated raw meat, and a failure of washing hands after handling this meat are means of transmitting the parasite. Now, the largest group of protozoans, which are the ciliates, have cilia all over most of their surface. The cilia allow the organisms to move, and in some genera, like Paramecium, the cilia can also assist in food gathering. Here you can see an example of Paramecium caudatum, which is also a ciliate. Now, Ballantidium coli is the only ciliate that parasitizes humans and causes dysentery. Ciliates have several highly specialized structures, and most ciliates have a well-developed contractile vacuole which regulates the cell fluids. Some have a strengthened pellicle. Others have trichocysts, which are tentacles that can be used to capture prey, or long stalks by which they attach themselves to surfaces. What is interesting is that ciliates can also undergo conjugation. However, unlike bacterial conjugation, in which one organism receives genetic information from another, conjugation in ciliates allows for the exchange of genetic information between both organisms. Now let's move on to discuss the fungi. Fungi are studied in the specialized field of mycology, and these are made up of a diverse group of heterotrophs. Again, many of these are saprophytes that digest dead organic matter and organic waste. Some can be parasites that obtain nutrients from the tissues of other organisms. Most fungi, like molds and mushrooms, are multicellular. However, yeast are unicellular. Here you can see a picture of the mycelium of a typical fungus on the bottom right. This is Aspergillus niger, which consists of a filamentous hyphae, which are cells of which can be multinucleate and separated from the spore-containing septae. Now, the body of a fungus is known as the thallus. The thallus of most multicellular fungi consists of a mycelium, which is a loosely organized mass of thread-like structures called hyphae. The mycelium is embedded in decaying organic matter, soil, or the tissue of a living organism. Mycelial cells release the enzymes that digest the substratum, which is the surface on which the fungus grows, and they absorb small nutrient molecules. The cell walls of a few fungi contain cellulose, but those of most fungi contain chitin, which is a polysaccharide also found in the exoskeletons of arthropods like ticks and spiders. All fungi have lysosomal enzymes that digest damaged cells and help parasitic fungi to invade their hosts. Many fungi can synthesize and store granules of the nutrient polysaccharide glycogen. Now, the hyphal cells of most fungi can have one or two nuclei, and many hyphal cells are separated by cross walls called septae. Pores in the septa allow both cytoplasm and nuclei to pass between the cells. Some fungi have septae with so many pores they are sieve-like, and a few lack the septae entirely. Many fungi can reproduce both sexually and asexually, but a few only have asexual reproduction. This asexual reproduction always involves mitotic cell division, which in a yeast occurs by budding. Sexual reproduction can occur in several ways. In one way, a haploid gamete unites, and their cytoplasm mingles in a process called plasmogamy. However, if the nuclei fail to unite, a dikaryotic or two-nucleus cell forms, and it can persist for several cell divisions. Eventually, the nuclei fuse in a process called karyogamy to produce a diploid cell. These cells, or their progeny, will later produce new haploid cells. Some fungi can also reproduce sexually during dikaryotic or diploid phases of their life cycle. Fungi typically go through haploid, dikaryotic, and diploid phases in their life cycles. And here you can see a diagram explaining the method of sexual reproduction in fungi. So let's walk through this. Here you can see that haploid organisms can maintain themselves by asexual spore formation. And this is shown in the beige part above, or budding. Alternatively, you can see below in the blue background that they may produce gametes that initially undergo plasmogamy, and they fuse their cytoplasmic portions. After several mitotic divisions of the still separate nuclei, the two nuclei will undergo karyogamy and fuse their nuclei to form a diploid zygote. The zygote then undergoes meiosis to return to the haploid state and produces reproductive spores. Now, fungi are important for a number of reasons, one of which is their role as decomposers in the environment. And with regards to health sciences, they are important as facultative parasites, meaning that they can obtain nutrients from non-living organic matter or from living organisms. Fungi are never obligate parasites because all fungi can obtain nutrients from dead organisms. Even when the fungi parasitize living organisms, they kill cells and obtain nutrients as saprophytes. Nearly every form of life is parasitized by some type of fungus. Now, some fungi can also be useful in the production of antibiotics that inhibits the growth or kills bacteria. Parasitic fungi vary in the damage that they can inflict. For example, fungi such as those that cause athlete's foot are nearly always present on the skin and rarely cause severe damage. However, the fungus that causes histoplasmosis can spread through the lymphatic system to cause fever, anemia, and death. Now, saprophytic fungi are beneficial as decomposers or as producers of antibiotics. The carbon and nitrogen compounds that they release from dead organisms contribute significantly to the recycling of substances in ecosystems. Parasitic fungi can be destructive when they invade other organisms, and these fungi have three requirements for invasion. They must have proximity to the host, the ability to penetrate the host, and the ability to digest and absorb nutrients from the host cells. Now, fungal parasites in plants can cause diseases like wilts, mildews, blights, rust, and smuts, and thereby produce extensive crop damage and economic losses. Fungal infections of birds and mammals are also responsible for extensive economic losses. Fungi that invade humans can cause human suffering, decrease productivity, and sometimes cause long-term medical expenses. Human fungal diseases are classified as mycoses, and these are often caused by more than one organism. Superficial diseases affect only keratinized tissue in the skin, hair, and nails. Subcutaneous diseases affect skin layers beneath this keratinized tissue and can spread to the lymph vessels. Systemic diseases invade internal organs and cause significant destruction. Some fungi are opportunistic, but they do not ordinarily cause disease, but can do so in individuals whose defenses are impaired, such as AIDS patients and transplant recipients who are receiving immunosuppressive drugs. Now let's move on to discuss the bread molds. The bread molds, or zygomycota, or also known as the conjugation fungi, have a complex mycelia that's made up of hyphae that lack septa with chitinous walls. The black bread mold, or Rhizopus, has hyphae that grows rapidly along a surface and into the substratum. Some bread mold hyphae produce spores that are easily carried by air currents, and when the spores reach an appropriate substratum, they can germinate and produce new hyphae. Now, sometimes short branches of the hyphae of two different strains, called plus and minus strains, will grow together, and the joining of hyphae gave rise to the name of conjugation fungi. Now, multinucleate cells form where the hyphae join, and many pairs of plus and minus nuclei fuse to form zygotes. Each zygote is then enclosed in a zygospore, which is a thick-walled resistant structure that also produces spores. Genetic information in zygospores comes from two strains, whereas that in hyphal spores comes from only a single strain. Now, bread molds do not typically cause human disease. However, Rhizopus is known to be an opportunistic human pathogen and can be especially dangerous to people that have diabetes that's not under good control. And here in the picture you can see a picture of the black bread mold, or Rhizopus nigricans. And you can see the sexual zygospores, which are the black spiny structures that are the result of joining and fusion of genetic materials at the tips of special hyphae side branches. And these zygospores germinate to produce a sporangium that in turn produces many asexual spores. Now let's talk about the phylum Ascomycota, also known as the sac fungi. This is a diverse group that contains over 30,000 species and includes yeast. The sac fungi have chitin in their cell walls and produce no flagellated spores. With the exception of some yeast which do not form hyphae, the hyphae of sac fungi have septa with a central pore. Unlike other fungi, they produce a sac-like ascus during sexual reproduction. Yeasts are included among the Ascomycetes, even though most yeasts have no known sexual stage. In species that reproduce both sexually and asexually, the asexual phase forms spores called conidia at the ends of modified hyphae. In the sexual phase, one strain has a large ascogonium, and an adjacent strain has a smaller antheridium. These structures will fuse, their nuclei will mingle, and the hyphal cells with dikaryotic nuclei will grow from their fused mass. Eventually, the dikaryotic nuclei can fuse to form a zygote, and the zygote nucleus divides to form 8 nuclei in each ascus. Each ascus forms 8 ascospores, sometimes releasing them quite forcefully. Here you can see the life cycle of an Ascomycete. Let's walk through this together. In the asexual phase, shown here in the greenish background, the spores called conidia are formed at the tips of modified hyphae. In the sexual phase, the mycelium that produces conidia also forms the gamete-producing structures antheridia, which is the male part, and ascogonia, the female part. After cytoplasmic fusion of those structures occurs, dikaryotic hyphal cells develop and interweave into an ascocarp, where the sac-like asci grow. In each ascus, the dikaryotic nuclei fuse to form a zygote, and the zygote nucleus divides into 8 nuclei. From them, the 8 ascospores form and are forcefully released. Now, many of the sac fungi are of interest to microbiologists. Nutrispora, for example, is significant because studies of its ascospores have provided important genetic information. Penicillium can produce the antibiotic penicillin, and other species of penicillium are also responsible for the color, texture, and flavor of certain cheeses. Yeasts, especially those of the Saccharomyces, will release carbon dioxide and alcohol as metabolic products of fermentation, and these are used in leavening bread and to make alcohol in beer and wine. However, there are also a number of sac fungi that are human pathogens. For example, Candida albicans can cause vaginal yeast infections. Trichophyton is one that's associated with athlete's foot, and Aspergillus can cause opportunistic respiratory infections. Species of blastomyces and histoplasma can also cause respiratory infections and spread throughout the body. Now let's discuss the club fungi, known in the phylum Basidiomycota. The club fungi includes mushrooms, toadstools, rusts, and smuts. The rusts and smuts parasitize plants and cause significant crop damage. In addition to having hyphae aggregated to form mycelia, the club fungi have club-shaped sexual structures called basidia. In a typical Basidiomycetes life cycle, sexual spores called basidiospores will germinate to form septate mycelia, and the cells of the mycelia will unite into dikaryotic forms. The dikaryotic mycelium grows and produces basidia, which in turn produce basidiospores. Some mushrooms, like Amantia, can produce toxins that are lethal to humans. Claviceps purpurea is a parasite of rye that produces the toxic substance ergot. The yeast Cryptococcus will cause opportunistic respiratory infections, which can be fatal if spreads to the central nervous system, causing meningitis and brain infection. Here we'll also discuss the phylum Deuteromycota. These are known as the fungi imperfecti, and these are called imperfect because no sexual stage has ever been observed in their life cycles. And without information on the life cycle, on the sexual cycle, the taxonomists are unable to assign them to a particular taxonomic group. However, based on their vegetative characteristics and the production of asexual spores, most of these fungi seem to belong to the sac fungi. Now we're going to move on to the phylae of parasitic helminths, in particular discussing the platyhelminths or the flatworms and the nematodes or roundworms. Now helminths or worms are bilaterally symmetrical, meaning that they have left and right valves that are mirror images. A helminth also has a head and a tail end, and its tissues are differentiated into three distinct tissue layers: the ectoderm, the mesoderm, and the endoderm. Now let's distinguish between flatworms and roundworms. Flatworms, or platyhelminthes, are primitive worms usually no more than 1 millimeter thick, but some, like the large tapeworms, can be as long as 10 meters. The flatworms lack a cilium, which is a cavity lying between the digestive tract and the body wall in higher animals. Most flatworms have a simple digestive tract with a single opening, but some parasitic flatworms, like the tapeworms, have lost their digestive tracts. Most flatworms are hermaphroditic, each individual having both the male and female reproductive systems. Flatworms also lack circulatory systems, and most absorb nutrients and oxygen through their body walls. Now, more than 15,000 species of flatworms have been identified, and this includes free-living and mostly aquatic organisms like the planarians, and two classes of parasitic organisms, the flukes and the tapeworms. Both of these parasitic groups have highly specialized reproductive systems and suckers or hooks by which they attach to their host. Now, with regards to the roundworms or the nematodes, these also share many characteristics with the flatworms. However, they do have a pseudocoelom, which is a primitive fluid-filled body cavity that lacks the complete lining found in higher animals. The roundworms have cylindrical bodies with tapered ends and are covered with a thick protective cuticle. They vary in length from less than 1 millimeter to more than 1 meter. Contractions of strong muscles in the body wall of these worms can exert pressure on the fluid in the pseudocoelom and stiffen the body. The pointed ends and stiff bodies allow roundworms to move through soil and tissues easily. There are more than 80,000 species of roundworms that have been described thus far, and they occur free-living in soil, freshwater, and saltwater, and as parasites in every plant and animal species ever studied. A single acre of soil can actually contain billions of roundworms. Many parasitic insects and plants, many parasitized insects and plants, and only a relatively small number of these actually infect humans. However, they can cause significant damage. Examples of roundworms that parasitize humans include the hookworms and pinworms, many of which live mainly in the intestinal tract. But a few, like the Wucheria, have larval forms that live in the blood lymph and can cause diseases like elephantitis. Now let's go over some of the parasitic helminths in particular. We'll start by discussing the flukes. There are two types of fluke infections that occur in humans. One involves tissue flukes, which attach to the bile ducts, lungs, and other tissues, and others involve blood flukes, which are found in the blood in some stages of their life cycle. Tissue flukes that parasitize humans include the lung fluke, or Paragonimus westermanni, and the liver flukes, Clonorchis sinensis, and Fasciola hepatica. Blood flukes include several species of the genus Schistosoma. Now, the parasitic flukes have a complex life cycle which often involves several hosts. The fusion of male and female gametes produces fertilized eggs that become encased in tough shells during their passage through the female fluke's uterus. The eggs pass from the host into the feces. When the eggs reach water, they hatch into free-swimming forms called myristidia. The myristidia penetrate a snail or other molluscan host, become sporocysts, and then migrate into the host's digestive system. The cells inside the sporocyst typically divide by mitosis to form radii. Radii, in turn, give rise to the free-swimming cercari, which escape from the mollusk into the water. These cercari can use enzymes to burrow through exposed skin, and the cercari can penetrate another host, often an arthropod, and then encyst there as a metacercari. When this host is then eaten by the definitive host, the metacercari exist and develop into mature flukes in the host intestine. And here on this picture you can see a picture of the Chinese liver fluke, which has been stained so that you can see the internal organs. It infests the gallbladder, bile ducts, and pancreatic ducts, where it causes biliary cirrhosis and jaundice. Here you can see the life cycle of the blood fluke, known as Schistosoma japonicum. This is the organism that's responsible for schistosomiasis. Unlike some flukes, this one does not have a radiase stage, nor does it enter an arthropod host. You can see here as we walk through the cycle that as the fertilized eggs leave the intestinal wall and pass outside of the feces of the host, the eggs will then hatch in the water. They become free-swimming myristidium that enter into a snail, which is known as the alternate host, and develop into a sporocyst. The free-swimming cercari can then enter the body, lose their tails, and encyst as metacercari. And then maturation of the adult worms will then occur within the liver and the hepatic ducts. Now regarding tapeworms, this again is a class of a parasitic flatworm. Tapeworms consist of a scolex or head end with suckers that can attach to the intestinal wall and a long chain of hermaphroditic proglottids. And here you can see how it can attach to the wall in the picture. This is the head or scolex of a tapeworm, and you can especially see the hooked spines and suckers used to attach to the intestinal surface. Now the hermaphroditic proglottids are actually body components that contain mainly reproductive organs of both sexes. The new proglottids develop behind the scolex, they mature and then fertilize themselves. The old ones will disintegrate and release eggs at the rear end. Among the tapeworms that infect humans are beef and pork tapeworms that are species of Taenia, dwarf and rat tapeworms that are species of Hymenolipsis, the hyatid tapeworm Echinococcus, the dog tapeworm Diploidium, and the broadfish tapeworm Diphyllobothrium. Although these different species display minor variations, the life cycle of tapeworms usually includes the following stages, in which you have embryos that develop inside eggs and are released from proglottids. The proglottids and eggs then leave the host's body with the feces, and when another animal ingests vegetation or water contaminated with these eggs, the eggs can then hatch into larvae, which invade the intestinal wall and can migrate to other tissues. A larvae can develop into a cysticus or bladder worm, or it can form a cyst. A cysticus can remain in the intestinal wall or migrate through blood vessels to other organs. A cyst can enlarge and develop many tapeworm heads within it, becoming a hydatid cyst. If an animal eats flesh containing this kind of cyst, each scolex can then develop into a new tapeworm. And here you can see a figure describing some of the different tapeworms that can affect humans. Here's the life cycle of the beef tapeworm, Taenia saginata. As you see, eggs can be passed in the feces along with the egg-filled proglottid. As the feces disintegrate, the eggs are then left on grass. Next, when a cow eats that grass, it can become infected, and then the eggs will hatch. The larvae will burrow through the intestinal wall, enter the bloodstream, and become encysted in the muscles as cysticerci. The cysticercus can then be found within the meat that may be eaten by a human, and when eating raw or undercooked meat or rare meat, the human can then also become infected. Now let's discuss adult roundworms. Most roundworms that parasitize humans live much of their life cycle in the digestive tract, and they usually enter the body via ingestion with food or water, but some, like the hookworm, can actually even penetrate the skin. These helminths include the pork roundworm, or trichinella, the common roundworm, or ascaris, the guinea worm, Dracunculus, and the pinworm, Enterobius, and the hookworm, Ankylostoma, and Necator. The life cycles of intestinal roundworms show considerable variation, and we're going to use the life cycle of Trichinella spiralis as an example. These worms can enter humans as encysted larvae in the muscle of infected pigs when undercooked pork is eaten. The cyst walls are digested with the meat, and the larvae are released into the intestine. They can mature sexually in about 2 days and then mate. The females will burrow into the intestinal wall and produce eggs that hatch inside the adult worm and emerge as larvae. The larvae can then migrate to lymph vessels and are carried to the blood. From the blood, the larvae burrow into muscles and encyst, and these cysts can remain in the muscles for years. And this same process also occurs in the pigs themselves, so that cysts are present in their tissues. And here you can see the life cycle of Trichinella, again showing that this is a cause of trichinosis, and you can see how larvae migrate or are taken in through eating undercooked pork, and then as stomach acid dissolves the cyst that's coating these worms, the worms can be released into the intestine, mate, get into the blood vessels, and then go into other parts of the body and encysted in the muscle. Now, whereas roundworms cause much of the tissue damage as adults in the intestine, some can cause damage mainly as larvae in other tissues. These roundworms include Wucheria bancrofti, which lives in the lymphatic tissue and causes elephantiasis; Loa loa, which can infect the eyes and eye membranes; and Onchocerca volvulus, which is the cause of river blindness, which infects both the skin and eyes; and Dracunculus mediensis, whose life cycle and symptoms are shown in Figure 11:21 in your book on page 330. Now, the larval roundworms are located in organs other than the digestive tract, and again they cause damage by the larvae. Here you can see an example of this. This is the life cycle of the roundworm Borreria bancrofti. This roundworm produces a microfilariae that cause elephantitis, which is a kind of chronic edema, especially in the legs and scrotum of affected individuals. And you can see here that this is transmitted through bites of mosquitoes, mosquito bites that then bore through the mosquito's stomach and thoracic muscles and develop into larvae. And as the larvae are then injected into the human through another mosquito bite, they then migrate to the lymphatic vessels and lymph nodes. And after they grow for about 6 months, the larvae can actually block the lymphatic drainage system, causing this elephantitis to occur. The last thing that we'll discuss here are arthropods as vectors. So arthropods make up the largest group of living organisms. As many as 80% of all animal species belong to this phylum. Arthropods are characterized by their jointed chitinous exoskeletons, segmented bodies, and jointed appendages that are associated with some of or all of the segments. Now, certain members of the 3 subgroups or classes of arthropods include the arachnids, insects, and crustaceans, are important either as parasites or as disease vectors. And if you check your book on table 11.5 on page 333, you can see a detailed list of diseases that can be transmitted by arthropods, listing the causative agent and the particular arthropod vector and the disease that it causes. Now, let's talk about some of these different arthropods, starting with arachnids. So arachnids have 2 body regions, and this includes a cephalothorax and an abdomen. They have 4 pairs of legs, or 8 legs total, and mouth parts that are used in capturing and tearing apart prey. And this includes spiders, scorpions, ticks, and mites. Now, infected ticks can transmit several kinds of human diseases. Some can